Good evening, everyone. This is Fahim, and this is going to be the final video that I upload that pertains to the applications of experimental design. Today, we are going to be looking at the last major piece of information that we need to finally build our own experiment and to run our own tests and analysis on different studies. And that's going to be my final project that I did. It's going to encompass every single thing that we've learned, which is randomization, multi-weight ANOVAs, different comparative tests, combine them all together so we can understand one last topic in this paper, which is going to be the randomized multi-weight factorial model. Let's get into it. This is the paper that I decided to do. I might have mentioned some of this information in a previous video, such as the environment that I conducted the study. The brief rundown is that there are two river systems throughout Savannah. One of them is here. You can see these different spots, which are in order of where I conducted sampling. 1, 2, 3, 4, 5, 6, 7. And there's one off the coast up towards here past Tybee Island that I wasn't able to capture in this image. But very similar river systems. The sampling sites go up the river and end towards the mouth. And the most important thing that we're trying to figure out is, can the river system that is past Tybee Island be used to substitute for the river in Savannah? The reason we want to do this is because studying river systems and estuarine environments throughout Savannah, they allow us to paint a bigger picture of nutrient export, such as nitrate concentrations, in and away from coastal areas. This is why we want to study these two systems. The problem is coming out to the system up here, you have to either take a boat away from the mainland and plan multiple day stays on the island, because it is an island where you're going, and it's a small island, so you can't take a plane. You can take a plane, but it has to be a pirate plane, which that already costs a lot. So we want to see, can we conduct the research on this river back here on the mainland? These are the different testing locations that I chose to do back when I did this research, and the exact distances are up here, but they're not too familiar. The reason why we want to ultimately use the multifactorial randomized design is because there's a lot of bias with this research that doesn't pertain to the methods used to analyze the nitrate. The methods that we use in the lab to determine what the concentrations are, those are robust. That's already been proven. All my references down here are basically proving that. Changing carbon cycles, oxygen degassing, different methods to test concentrations in the lab, those are all fine. That's not what we're interested in. We're interested in whether or not we can compare the two systems together. And are there bias while taking those samples? And there are. That's why we're trying to randomize this study. One of the issues is that the actual sampling process occurs on multiple days, 2 days to be exact, at different tidal stages. The biggest problem that you're going to face is that it's tiring. You're on the open water for half of the day. When you get down to station 1 and once you finally finish with station 7, that is exhausting. You might not be paying attention to what you're doing as much, which is really bad to say because you really should as a scientist, but you'll see one of the outliers is due to that. I wasn't being very careless and I took a measurement I wasn't supposed to be taking. That's one reason we need to randomize the experiment, to reduce that bias and error, as well as the other nuisance factors that we'll be getting into. This is just more etc. that we don't need to concern ourselves with. Data collection plan. And what about the results? What are we doing exactly? There are multiple factors that we are considering. These factors are the day, the 2 days we went out to conduct the sampling, the 2 rivers, there's 2 days, 2 rivers, 7 locations. In this study, we actually learned when I finished this paper that the locations didn't make a difference. Those are a non-significant factor. We could leave those out, which is good. That is the only thing that remains same, and that's what should remain same. The actual locations, which we took each sample, were the exact same way. This is where we start getting into some of the more logistical parts. The river that runs through Savannah and the mainland, this system here is both fresh water towards the mainland and ocean water. It has high salt concentrations and lower concentrations of nitrate. As you move towards larger bodies of water, your concentrations of nitrate become less, opposed to when you have the river running through the ocean, which is where Sapelo Island is, where the other testing was conducted, that's essentially all ocean. It's very low concentrations of nitrate, which is what we observed when making the data sets in the table, and which was one of the reasons we, of course, randomized our study. When we did our residual analysis on the entire data set, it's very skewed. You're able to take a look at the p-value for our data sets. It's very low. It's significant. It looks like a transformation is going to be necessary. There's a bit of variance as you move towards higher fitted values throughout the residuals of the analysis. Looks like there's a couple of outliers in time series. And time series is also very important for this autocorrelation because we do know the run order. The run order is right here. There was a specific time that we took all these measurements at a specific distance. So we do have to transform the data. The square root transformation was most appropriate. It definitely helped the residual analysis. So it's a very fair transformation. Histogram, it's decent. We did not omit the outliers, which we did with Cook's D test on Minitab. Of course, you don't ever want to omit outliers. You want to recognize them, but they are points that we chose to include. And this is where the bias came in. When I was taking these measurements in the ocean, one of the other factors was depth. You have to take a measurement at the surface for concentrations and at the bottom. When you put the bottle towards the bottom that we used to get the bottom measurement, the Niskin bottle, which is the name that it's called, captured some of the sediment, which has high concentrations of nitrate inside of there, which is what you're going to observe at these points right there, which is unfortunate. But this is how you know it's randomized, because I remember specifically we caught it at this point right here, the bottle, which had the higher concentration, and this randomized. It's towards the middle right here, which is good. That's what exactly we want to see. Ultimately, what we wanted to check out and what we wanted to conclude was what happens if we do a full factorial model in Excel, because you can do this in Minitab, but the web app is just so slow with this that I decided to do this in Excel. Excel moved a lot faster, where we did Cohen's D test with the replaced value of the groups for the appropriate mean factors and standard deviations. And we found that the most significant factor is going to be the river itself, which makes sense. The river, they are two completely different systems, was what we concluded, and they cannot be used as surrogates for one another when conducting this research. Again, I did go over the specific outliers that I conducted or recorded with the Niskin bottles, and the actual multifactorial experiment that I conducted was randomized. I attempted it in Minitab, didn't go too well, finished it in Excel. That is the very quick and dirty rundown. I cannot go any longer talking about this because not only will I bore you guys, but it's a long paper. There's a lot of logistics you have to understand, but it's all here. And it's something I'm proud about when completing this course. So thank you guys again, and I hope this gave you some insight on what this course was about.